get a gradient again of SOG protein throughout the embryo. So here, SOG protein is expressed, or it's produced in the most ventral regions of the embryo, and the concentration of SOG protein in the cytoplasm will migrate away from this source, typical to what we see with the morphogens. DPP protein is expressed mainly in the dorsal regions, and so its concentration will migrate down. Okay, so it turns out that SOG and DPP protein interact, and they actually physically bind to each other. So when SOG binds to DPP, it basically prevents the DPP protein from binding to anything else, to binding to any other receptors, so it's bound up with this SOG protein. And the SOG protein is still being produced, and it's binding to more DPP as it's migrating up, so it's thought that the SOG and DPP proteins kind of end up being escorted back up towards the dorsal regions of the embryo. Okay, so you have differential expression of SOG and DPP, and you also have the prevention of DPP activation because it's basically stopped up by this SOG protein. And the SOG protein is bringing that DPP protein back up to the dorsal side. Okay, so then this helps to define those regions of the embryo that I told you in the previous amnioserosa, the dorsal ectoderm, the neuroectoderm, and the mesoderm. Okay? Because here, the mesoderm is in the most ventral region. There's basically SOG is produced here, but there's no DPP. So that pattern of high SOG, no DPP is defining this as mesoderm. As you go more dorsal of the embryo, you have a region that has higher SOG protein, less concentration of SOG protein, maybe a little bit of DPP protein, and this is signifying the region that will become neuroectoderm. You go further up, and at this stage, you have SOG and DPP together as a complex, and there's expression of this other protein called tolloid, TLB, that basically chops SOG up, so it releases that DPP that it was bound to. And so that allows the DPP to be active in this region of the embryo, and that becomes more dorsal ectoderm. And then if you go even to the most dorsal end of the embryo, you have the highest levels of DPP activity. And so high levels of DPP activity signifies the amnioserosa region. Okay? Okay, so again, you see kind of different proteins in different concentrations in very specific areas of the embryonic body, and that helps to define their future fate. That's a lot. That's a busy slide. So we'll stare at it for a while as we take a break. This is 3:45, so let's take a 5-minute break, try to get our head around this slide, and then we'll go forward.
I forgot where I put the green ball. Huh? No, I'm not going to pause it because it has to process, and it might take a while to process.
One was, where is the TLB actually produced in the embryo? And so it's hard to tell from this diagram.
Move it closer.
It's mainly produced in the dorsal hemisphere. Okay, so it's around here, but it's not produced in the ventral region. Okay, SOG is produced in the ventral region. And it migrates up, okay, or diffuses up. DPP is produced at the dorsal region and it diffuses down, but then they meet in the center. And when they meet in the center, DPP and there's more DPP coming down, but I think the way that it has to work is that there must be more SOG production than DPP production because there's more DPP SOG complexes found in the the middle region of the embryo. Okay, so you have these complexes, and then when these complexes are migrating towards the dorsal region, they meet up with TLD, and TLD is a protease, and so basically it eats up the SOG and releases DPP, okay, and it kind of keeps the concentration of DPP in the dorsal side of the embryo. So as I told you, the DPP is produced from the most dorsal cells. And so you have the production there, and then you have this kind of trapping of the DPP in the dorsal regions of the embryo, so you get really high DPP activity in the most dorsal area of the embryo, and you don't have really any DPP activity in the ventral region. And in between, you have kind of this mixture of activities. Okay, and so this region is more ventral. This one where there's complexes, or certain concentration of complexes would become neuroectoderm. This one that's got kind of some DPP complexes and some that are releasing DPP may become more dorsal ectoderm. And the areas that have more free DPP become amnio, serosa, and just the most dorsal regions of the embryo. Okay? So again, we see how different proteins interact and how changes in their activity in local areas result in very different anatomical regions, so a different body plan. Yes.
I have a question.
Mm-hmm.
Why doesn't the DPP just stay up there?
Why didn't it just stay up there?
No. It just doesn't.
Well, okay, so it's a good question. Why didn't it just stay up there? So how could you keep a protein in a certain location in a cell in a body? You could have it anchored there by extracellular matrix protein, or there could be just active transport of that protein, to keep it there, similar to the way the maternal genes are localized in one area, but it doesn't seem to, none of those mechanisms seem to apply to DPP. So I don't know. I know it seems very complicated, right, to have this process to just keep something there, but that's just, okay. And now, I always feel like I'm doing a lot of hand waving when I give answers like that. But I just found a paper this morning that was published on January 1st of this year that actually talks about the specific cytoskeleton and actin and dynin proteins that bind only to bigoid and keep bigoid in the anterior region. So it's still being worked out what's the active mechanism that makes a protein stay in one place versus another place in the Drosophila embryo. So yeah, it's still really knows. Okay, so the ultimate effect of the dorsal gradient is that you have now a true dorsal region in the embryo and a true ventral region. And you start to get the expression of specific genes that start to develop into some of the germ layers. So for example, in the ventral regions of the Drosophila embryo, they start to express genes that are fated to become mesoderm. And these are the cells that will develop into the gut. And so if you look at these genes, so here example is, I think this is twist, that's being stained by immunohistochemistry. And you look at where these cells that express twist are located at different times, and you can see they're starting to gastrulate, they're starting to undergo a process called invagination, where the gut is basically